Thank you, Federico. Thank you for having me this afternoon. I've been supporting this adventure of the Max Planck groups in Colombia in the backstage, and today I've given the opportunity to tell you something that we have learned with a particular model that we've been working. Next, please. Do I change it with this? We have been working in the last 2 decades with this parasite, leishmania. It's a parasite of public health importance in Colombia. In Colombia, we also have an interesting distribution of these species because the Andes somehow separates the subgen, which is towards the north of the America, and the other subgerms that is to the south of the Andes. In this particular case, you're looking at Leishmania brasiliensis, which is the species we've been working with. And we have set in our lab an in vitro life cycle to ask some questions in relation to the physiology of the interaction between the parasite and the macrophage, an immune cell which happens to be the host where this parasite reproduces. This form that you have here is the Promaster. This is a free flagellated form that is found in the intestine of the insect that normally carries the disease in the wild. This is a macrophage which has been recently infected. You see that the shape of the parasite is nearly kept, but the parasite goes to a transition to the differentiation stage of the amastigote. If you go with me. This is a macrophage around 72 hours post-infection, and the parasite changes in size, changes in morphology, and also in the conditions where it normally lives. As this free form is normally found in the insect, it encounters a pH of about 6 to 8, and this range of temperature. But when it gets into the macrophage, the parasite is exposed now to a very low pH. In terms of the cellular niche, this is more or less hell. That's where Leishmania thrives, and somehow it's really well adapted. So our questions are around how is that this parasite tolerates this really big change of pH, and what mechanisms allow them to do it. We started a long time ago. Actually, I saw Luisa was here. She was the first one that started this work as a master's degree student in my lab. When we started, we knew very little about the physiology of the parasite. It's been a lot of research in terms of immunology, but no real research in terms of the physiology. And we assumed that if we want to understand this relationship and to come up with real solutions, we have to understand what we're dealing with. In the middle of the 80s, a group led by Silverstein had established that there was a pump, a proton pump, that used energy in the form of ATP to create a gradient that was utilized by transporters of glucose and proline to make sure that the parasite had its nutrients. The actual pump was described much later by Marcela and Docampo, and these tend to be, this is a PATPase, an ATPase, a P-type ATPase, which has certain features that related with the sodium potassium ATPase, that is the main pump in the animal cell. The function of this pump creates a problem, and is that it extracts protons in terms of charge, a positive charge, outside the parasite. And if this happens without any help, the pump will stop. And if the pump stops, the parasite cannot enter the nutrients it needs. So it has been suggested by the time that there has to be what is called a chloride shunt, some molecule that allow the pump to continue its functioning without building the electrical gradient involved. So our first question was, what was this shunt, if there was any there? And we went there looking for a chloride channel, because that was the theoretical assumption that we derived from the literature. Sorry. Initially, the first experiments we did very long time ago were kind of a very long way. We took all the mRNA of leishmania, initially amazonensis, then brasiliensis. We inject that total mRNA into all sites, and then we record it. And then was when our collaboration with Walter's tumor started, because Luisa went to his lab and started doing some of the recordings in a species of amphibia, Xenopus oocytes, that is well established there. Then we had the opportunity to study the biodiversity here. We showed that we could use also Bufo marinus, which is a local native species. Well, we cannot longer use it because our bad use of it actually endangered it. So we're not longer allowed to do the experiments we were supposed to do. What we did here is that we injected total mRNA of leishmania into all sites, and then we did with voltage clamp, two electrode voltage clamp recordings. And what you have here is the recording of a control oocyte, a recording of an oocyte that was injected just with a buffer where the mRNA was diluted, and a recording of the injection with mRNA. And what this is showing us is that there is voltage-dependent current that happens to be driven by chloride. And with this information, we knew that we were dealing with a particular molecule that belongs to a family which is called the CLC family. At the time, the Leishmania brasiliensis genome was published, so then we switched into looking for the potential candidates for CLCs in these parasite. The CLCs were first described by Christopher Miller, and I like this quote because they have been really neglected in terms of study. They seem to be the ones that clean the party after the very speedy potassium and sodium channels do the action potential and do the whole thing that allows the brain to function. But the ones that keep the system functioning, or at least some of them, are this family of molecules. When they were first described, they were described as chloride voltage dependent channels. Here is just a recording, and something that took the attention of the people is that these proteins have two pores, which were not the usual model previously accepted for an ion channel. We assume that the ion channel has only one pore, and through that pore the ions permeate. Not in the case of the CLCs. They do work as homodimers, and each dimer has its own chloride pathway. Later, this same group, Christopher Miller, with Akadi and some other collaborators, were able to clone the E. coli, one of the CLC of E. coli, and to their surprise, they didn't find a chloride channel. And this is quite interesting because in biophysics, you are either a channel or an exchanger. And this family has the two ways of moving ions, if you want. So in this case, you have a molecule which is not voltage dependent, but it's triggered by pH and functions not like an ion where you just have in one direction the flow of one ion. Here you have a kind of pump in terms that the electromotive force of one ion will make sure that you can pump the other one against its gradient. And in this case, this is a chloride, a proton chloride exchanger. When we start studying the CLC family in leishmania, we found 4 candidates which appear to be exchangers with our bioinformatics studies. And it's interesting because in the mammalian tissues, the channels are expressed on the membrane, whereas the exchangers are expressed always intracellularly. And in the case of the leishmania, one of them, which is the one that I'm going to talk in more detail, it's relatively close to the only CLC that yeast has and that we know already is expressed in one compartment inside the cell, which is the Golgi apparatus. And surprisingly enough, in that location, what this CLC is doing is guaranteeing the proper expression of some transporters, in particular transporters related with the transport of ion. What we have learned so far about this particular one is that it is transcribed. In other words, the leishmania appears to be using this gene and is differently transcribed. This will be the Promaster, the free living form that I present you initially. And these are different times, a period during which this promastigote should transform into the amastigote. This is another form, an amastigote that we can generate without the macrophage, but in our hands at least, they do not behave the same, so we call them amastigote-like. The parasite expresses the gene in all these stages, but we see a very interesting increase during the first hour post-infection, as if this putative exchanger was indeed important for adaptation into the macrophage later on and towards this acidic pH. These, it's not, I don't know if it's well seen there, but these are promastigotes. And what we did here is with a specific antibody that was designed by one of the PhD students of the time, Michel Garcia. We found that it's located towards the plasma membrane, but also in very tiny vesicles that are close to the membrane. This is interesting because CLC3 and CLC5 in mammalian tissues have exactly the same location, and there they regulate the expression of different transporters in the plasma membrane. We then collaborated again with Walter, and in this occasion, Michel Garcia and Jenny Lozano and Julie Buitrago had the opportunity to do their half a semester there or a semester in their lab. And what we did here was that we cloned this, the gene that expresses this protein. We inject all sites again, and we did similar recordings as the ones previously mentioned. And what we found here again was a current. Here we have recordings in pH 5, pH 8.5, and 7. Should be a point there and not a column. My apologies. And what we found here is that had the previous features that we have already described, which is a protein that induces a voltage current, but it did not appear to be particularly pH sensitive. Something that struck our attention at the time is that the oocytes that were injected with this with the construct changed volume, and somehow that suggested to us that this molecule was related to the regulation of this parameter in the parasite. We also tested the possibility that other anions were permeated by this molecule, and this is because this is not a very selective either channel or exchanger. The idea is that we call them chloride channels because that's the anion that we normally find in big amounts in most tissues. But there had been a description in plant cells, and I mentioned here that if you want to study leishmania, you have to assume that this is half animal, half plant, because the physiology is, the protozoan physiology is kind of different. It's not exactly, probably protozoan physiology, but we don't accept that term yet. What we found here is that nitrate was also permeable, and this is very interesting in the context of leishmania, because nitrate is one of the radicals that is produced as a secondary product of nitric oxide production by the macrophage. Once the parasite is placed in hell, not only the pH is low, it's also full of degrading enzymes there, and many of the oxygen bursts that the macrophage can actually launch to theoretically defend the host against this invader. To understand a bit better the function of these, I mean, we already know it appears to be voltage dependent. Chloride and nitrate also permeate, but to understand exactly what the function is in the parasite, so we created lines where this molecule was silenced. To do this, we follow the work of Elisabetta Ulu, she was the guru of silencing in leishmania. And in this case, fortunately, we were working with the right species. In the case of leishmania, it's interesting because only the subRNA gene has silencing machinery. The other genes somehow have lost it or have a different kind of machinery that hasn't been described. So what we did, went to a very complex cloning procedures to silence a line. This was the PhD work of Cristina Zapata, and she got a leishmania line that theoretically expresses less amount of this protein. And the first thing we found with this line is that it grows less than the wild type or the line that is only transfected with the plasmid, the bacterial DNA that we use to transform these parasites. One of the interesting things is that we went to look for changes in volume because of the previous data. Leishmania is an eukaryotic cell. As any eukaryotic cell should be tested with a hypotonic challenge. It should increase in volume and then gradually should decrease in volume, should do that. All eukaryotic cells do that. This is our experiments where we challenge the control leishmania and the transfected leishmania, and the control leishmania was able to increase volume and decrease to its basal level, but the transfected leishmania was not. So this was the initial proof that physiologically speaking, this molecule is associated with a regulation of volume in leishmania. Then we did something because we had the system, and we look at the morphology. This parasite it's different. I'm going to come to this data later if the time allows. But to our surprise, when we infected with this transfectant, this is a 24 hours infection, but this is a 96-hour infection. I don't have it, no, here. And to our surprise, after 96 hours, the macrophage killed the parasite, and that shouldn't happen. The way we do the experiments is for the parasite to survive. And if you look carefully, the morphology of the parasite is promastigote. In other words, the parasite cannot differentiate from the free-living form to the intracellular form. And somehow the volume is a signal for the parasite to be able to differentiate. Coming back to this data, when we look at this data after having these experiments, we realize that the differences in morphology here could be interpreted as that the parasite was not maturing properly, that this form that we had here was not the fully flagellated form that is ready to infect, but the previous one that we call the procyclic form. Okay. So I'm going to finish because I've got the red light. So we think we found the shunt. And in the promastigote, our molecule should be doing this. But in the case of the amastigote, we have a more interesting theoretical explanation that we should follow. And is that, in this case, if this exchanger is capable of permeating nitrate, then one could envisage a model in which the macrophage is producing nitric oxide. Nitric oxide is converted into nitrate, and then the parasite has a pump to detoxify it from nitrate. And the problem is that it has to deal later with the pH. But looking in the literature, we found some work done by a French group that suggests that nitrate is the signal that leishmania requires to remain a mastigote. So in this case, it works as well, but it would not be detoxifying the parasite from nitrate. It would be keeping nitrate as a signal to maintain in that particular stage, helping the parasite to deal with the pH pressure that means living in the niche of the parasitophorus vacuole. I'm just going to list here the people, most of them students, masters or PhD in my group, and finally the people supported this work. Thank you.